Hello children. So in this video we are going to talk about the last step of aerobic respiration, that is electron transport system, which is also known as electron transport chain, or even it is also known as respiratory chain. Right? And it can also be abbreviated as ETS or ETC, that is electron transport system and electron transport chain. Now, during aerobic respiration, this is the last step of aerobic respiration, and we have studied that aerobic respiration, it utilizes oxygen, but the glycolysis step, the acetyl reaction step, the Krebs cycle, and all those things, we have seen that there is no utilization of oxygen. So actually the oxygen that is going to utilize is during electron transport system precisely. So here oxygen utilization is going to take place, first thing. Okay, and why this electron transport system is actually required? Well, it is required to, if you remember in previous glycolysis step or the Krebs cycle or the acetylation reaction step, in all those certain high energy molecule like NADH or FADH2, these were being formed. So what is the fate of all these? Well, these molecules, they are going to ultimately give their electron and proton to this electron transport system, and ultimately we will see the ATP production is going to happen. So that is why this electron transport is necessarily going to be there in the case of aerobic respiration, right? Another thing, that where exactly this electron transport system is present. So the site of electron transport system, or the location of ETS, is actually the inner mitochondrial membrane. Okay, inner mitochondrial membrane. Okay, now this electron transport system, it includes two things in firstly. Okay, let us just write it down. So it includes two things. The very first one is presence of certain complexes. Okay, so what are these complexes? Let us discuss. There are going to be in total 5 complexes. The very first one is NADH dehydrogenase, also known as ubiquinone oxidoreductase, right? Another complex, the second complex, is succinate dehydrogenase, which is also known as FADH2. Third complex will be cytochrome BC1 complex, which is also known as ubiquinone cytochrome C. Fourth complex is going to be cytochrome C oxidase, which is also known as cytochrome AA3 complex. And And the last complex, that is the fifth complex, it is ATP Synthase, also known as FOF1 complex or FOF1 particle, or basically the oxysomes which are found in the inner mitochondrial membrane. Okay, so these are the five complexes that are present in or make the electron transport system. We will see how. Now, one thing that you need to remember, children, that among all these, like out of five, first, third, fourth, and fifth, these four complexes are going to be embedded in inner mitochondrial membrane. They're totally fixed or embedded in this inner mitochondrial membrane. While the second one, this one, the second complex, it is the one that is present towards the outer side of the inner mitochondrial membrane. Okay, so this is what you need to remember about complexes. Now, another thing, I told you first thing is complexes that is present in electron transport system. The another thing that is present in electron transport system is presence of certain electron carrier. So, certain electron carriers will be there. So, what are these electron carriers? The very first one will be something known as FMN. The full form, let me write it. Flavin mononucleotide. Okay. Another electron carrier will be ubiquinone or coenzyme Q. Third will be something known as iron sulfur protein. This FES or iron sulfur protein will remain associated with two things. One is FMN and another one is cytochrome B. We will see how. Okay. Now, the fourth one will be cytochrome. Now, this cytochrome can be of different types. There can be cytochrome B, C1, C, A, and A3. Try to remember in this sequence itself, because in this very sequence they will be present in the electron transport system. So if you remember in this sequence, it will be easier for you to put all these in electron transport chain as well. Okay. Now, these are the electron carriers that are present in electron transport chain, and among these you need to remember that the second electron carrier, that is a ubiquinone, also known as coenzyme Q, is a mobile carrier. So it is not going to make up the part of any complex. It is present in between the complex and it is a mobile carrier. Okay. Just like that, your cytochrome C is also a mobile carrier. So this one will be a mobile carrier as well. Another thing that you need to remember, that cytochrome is actually a kind of heme protein. Sometimes they just ask this question in competitive exams. So it is actually a heme protein. All the carriers are proteinaceous only, but this includes heme group as well. So heme protein. Okay. So electron transport system, we have seen, has two things. One, the presence of complex, and second, the presence of certain electron carriers. And the site of or the location of this electron transport system is inner mitochondrial membrane. So let us just draw this electron transport chain and let's see what is going to happen there. So let's say this is going to be the outer mitochondrial membrane. Here is outer mitochondrial membrane, okay? And this here is going to be your inner mitochondrial membrane. Let me just provide a bit clear. So this whole is your inner mitochondrial membrane. Okay. Now, between outer mitochondrial membrane and inner mitochondrial membrane is present intermembranous space. The space between two membranes is intermembranous space. Okay. And this whole is your matrix of the mitochondria. So towards this side is matrix and towards this side is outer mitochondrial membrane. Okay. And if you remember, I've just told you that electron transport system is a part of your inner mitochondrial membrane. So this is inner mitochondrial membrane. So it includes 5 complexes. So let us just draw those 5 complexes first. So they're going to be the very first complex. There it is. First complex, right? Then third complex, I'm drawing third, not the second one. So they're going to be third. And then the fourth one. And ultimately the fifth one, that is your ATP synthase, and it includes FOF1 particles. So this is your FOF1 part, right? So if you remember, we have just talked about that among these 5 complexes, first, third, fourth, fifth are embedded in inner mitochondrial membrane, but second one is not, right? So I have made this first, third, fourth, and the fifth one. So this is the fifth one. Okay. So second one is actually present here. So here, this is the second complex. Okay. So first, second, third, fourth, fifth, 5 complexes. Now, I told you what is going to happen in electron transport chain. Ultimately, NADH and FADH2, which were formed during glycolysis and Krebs cycle and all, they're going to donate their proton and electron to this electron transport system, and ultimately ATP production is going to happen. So in this electron transport system, we are going to see how ATP production is going to happen. Okay, and so just let us start. So first of all, what is going to happen? NADH is going to come. So we are going to start with NADH first, right? So NADH will come here. NADH plus H plus, which was formed during Krebs cycle or glycolysis and all. And what is going to happen here? It is going to donate its electron and proton and ultimately going to get converted into NAD plus. NAD plus. So here what is happening? The electron, it will release, this NADH will release electron, and this electron, it will release 2 electron, and these electron are going to picked up by something known as FMN. Okay. Now, what is going to happen? Since these electrons are being picked up, this FMN, so of course it is going to donate the proton as well. This NADH is releasing proton as well. So from here, the protons are also entering inside this first complex. So these are the protons. Okay. Now, eventually what is going to happen, this FMN will further, since it is the electron carrier, it is going to take up the electron and again pass on to another one. From this, the electron will be passed on to, the electron will be passed on to another electron carrier, that is your iron sulfur. Just write here. Electron is going to pass on to iron sulfur. Okay. Now when this electron will be passed on from FMN to iron sulfur, what is going to happen? What is going to happen? Eventually, these protons which were actually given out when NADH was releasing the electron, these protons are actually entering or leaving this first complex and ultimately entering towards or to the intermembranous space. Right? Now, this iron sulfur, which is a part of this first complex, is having this electron. So this extra electron, so this whole first complex is feeling kind of very negative, since it has all the extra electron. It has all the extra electron in it, right? So what is going to happen? What is going to happen? This first complex, in order to neutralize that, it is going to pick. It is going to pick certain protons from the matrix. In order to neutralize that negativity. So here the protons are picked up by the first complex. Now eventually these electrons, which were the part of iron sulfur, now will going to get this iron sulfur is going to release this electron, and ultimately this electron will be passed on to the mobile carrier, that is ubiquinone. Now since all the electrons, I've shown only passing of one electron. Eventually the two electron is getting passed on, but at one time, like at a single point, only one electron is getting picked up and passed on, like this. So I'm going to show only passing of one electron, but you need to remember that two electrons are getting passed on. Okay, so ultimately then these electrons are going to get released from the first complex to the ubiquinone. This whole system is no longer feeling very negative kind of thing, right? So these electrons which were picked from the matrix are now ultimately going to get released to the other side of the membrane, that is towards the intermembranous space. So here two more protons are going to get come. So ultimately, how many how many protons are getting released through the first complex? I can say in total 4 protons. 2 protons from NADH, which were released from NADH, and 2 protons which actually picked up by the matrix. So ultimately 4 protons here. Now, this ubiquinone has this electron. Okay. Now, ubiquinone is just not an electron carrier, it is a proton carrier as well. So, whenever it is going to pick up the electron now, it is also going to pick up the protons as well. And now these protons are again will be pulled up by matrix only. So, when it is going to pick all the protons from the matrix, it is this ubiquinone is going to get converted into its reduced form, that is ubiquinone UQH2 reduced form. Okay, so ultimately what is going to happen? Again Again, this ubiquinone, since it is a mobile carrier, will move in between first and third complex, and ultimately this ubiquinone is now going to release its electron and will give this electron to another carrier, that is cytochrome B. That is the part of third complex. Now cytochrome B will eventually give this electron to iron sulfur. If you remember, we have talked about iron sulfur is associated with FMN as well as cytochrome B, right? So from cytochrome it is going to get passed on to iron sulfur, and from iron sulfur it is going to passed on to cytochrome C1. So these electrons are getting passed on from here to here, from cytochrome B to iron sulfur to cytochrome C1. When ubiquinone is now releasing its electron, and it has just taken up the proton in order to neutralize this electron which was taken from iron sulfur. So when it is passing or releasing the electron, this ubiquinone will eventually also release this proton, and again towards the intermembranous side only. Okay, but this u proton, or I should just draw it like this. These protons are actually getting released. Just wait a minute. Right. So this electron of these protons which are picked by ubiquinone H2 are going to get released towards the intermembranous space, but first they will enter inside the third complex and ultimately getting released towards the intermembranous space. So two protons are coming from here, and since again these electrons become the part of third complex, it again is going to feel that negative vibe, you can say, because of presence of those extra electrons. So again this third complex is going to pick the protons from the matrix, and eventually when electrons are getting released or given out again to another complex, these protons are going to get released towards the intermembranous space. So again, through third complex, total 4 protons are going to get released towards the intermembranous space. Okay, now when cytochrome reaches C1, it is going to release this electron. It will be picked up by another electron, or you can say another mobile electron carrier, that is your cytochrome C. If you remember, we have marked it earlier as well, right? Now, this cytochrome C will eventually pass on this electron to the fourth complex, and in the fourth complex there are many electron carrier present, so let us just draw them. They're going to be copper A, cytochrome A, cytochrome A3, and copper B. So these are the carrier that are present in the fourth complex. So ultimately the electron will be passed on from cytochrome C to to copper A, then to cytochrome A, then cytochrome A3, and ultimately to copper B. Okay. Again, this complex is failing negative because of presence of all these extra electrons. So ultimately this will also going to pull up the protons from the matrix, and when this electron eventually be given out to the matrix side, we will see who is going to pick it up. But again, when this electron is going to get out of this fourth complex, ultimately what is going to happen, these protons will be released towards the outer side. So during fourth complex when electron transfer is happening, please remember that only two protons are going to get released towards the intermembranous space. Why? Because here what is happening, the ubiquinone was a proton carrier. Okay, when it is accepting electron, it is going to have some protons as well. But cytochrome C is just electron carrier, so it doesn't require any proton in order to neutralize that negative vibe. Okay, so it doesn't require the pulling of proton from the matrix, right? So during fourth complex, or through the fourth complex, you can say only two protons are going to get released in the intermembranous space. Okay. Now eventually when this copper B is going to release this electron, it will be picked up by the atmospheric oxygen that is present inside the matrix region, that we inhale during breathing only. Okay, so this oxygen is that oxygen only, and this electron is picked up by this half oxygen, and with this it also combines with two protons, which is again coming from the matrix only, and ultimately form a molecule of water. So two protons, two electrons. I have shown you, again I am telling you, I have shown the passage of only one electron, but ultimately the NADH has released two electrons. So ultimately here two electrons will be accepted by the half oxygen and two protons from the matrix, and one molecule of water is formed. Okay, so this was the passage of your the protons, or the protons that were, or the electrons that was released by NADH, and it includes the complex first to third to fourth. Now what is going to happen? What is going to happen with the FADH2? What about FADH2? Now let us talk about FADH2 now. So, if you remember, when we were writing the names for all the complexes, the first one is NADH dehydrogenase and the second one is succinate dehydrogenase. Okay, so since first one was NADH dehydrogenase, so NADH is going to give its electron protons to the first complex. But if you remember from the Krebs cycle, the FADH2 is formed at one single step where you can say where the succinic acid is getting converted into fumaric acid, right? So here only during single step the FADH2 is getting formed during Krebs cycle. So at this step, uh, as soon as the FADH2 is going to form, this FADH2 is going to release its electron and proton, and it will give it to second complex, that is succinate dehydrogenase. That is why the name succinate dehydrogenase, because the step was the succinic acid getting converted into fumaric acid. Okay, so it is going to donate its electron as well as proton to iron sulfur that is present here. Okay, and this iron sulfur is ultimately going to again release the electron, okay, and ultimately giving it to this ubiquinone that is present in the inner mitochondrial membrane. So ubiquinone, and this ubiquinone again, just like that, and that is going to get converted into ubiquinone H2, that is a reduced form of the ubiquinone, by accepting electron as well as by accepting proton from the matrix, right? Just like that we had discussed earlier as well. Now this UbQH2 is again going to donate its electron to cytochrome B of the third complex, and when it is doing it, it is going to release this proton over here. And again, when protons are getting released here, and eventually this electron is passed on from ubiquinone H2 to cytochrome B, these proton, which was accepted by ubiquinone, is going to release inside the intermembranous space again, just like what we had discussed in the case of NADH. And since this electron is now the part of this third complex, again it is going to feel that negative vibe and will pull some proton from the matrix, and eventually when release it and give these electrons to cytochrome C, so it will no longer require that proton, and ultimately those protons will be released towards the intermembranous space. So again here 4 protons are getting released. Just like that, the cytochrome C is going to accept that electron again, and this electron will be passed on to fourth complex in copper A, cytochrome A to cytochrome A, not A3 and copper B. And ultimately, this electron will again be passed on or accepted by the final acceptor, that is oxygen, and it is going to form water molecule. So again here, just like that, 2 protons are going to get released in the same way that what we have talked about during NADH, electron transport chain, right? So eventually what is going to happen, the final acceptor in the case of electron transport chain of this electron is oxygen, and that is why this electron transport chain, it is a part of aerobic respiration that utilizes oxygen here. Now we have seen the electron transport chain or the electron transport system through the NADH and FADH2, but one thing I told you that here what is going to happen, ATP production is going to happen, that we haven't seen it yet. So how ATP production will happen? So see what is happening. See here in the intermembranous space, in mitochondria, in intermembranous space, the proton concentration is getting very much high. All the protons are getting released in the intermembranous space. From the matrix, they are getting pulled up from the matrix only. So here in the case of matrix, there is low concentration of proton. While here in the case of intermembranous space, there is high concentration of proton. So a proton gradient is being formed. A proton electrochemical gradient is being formed. Here the higher concentration, here the lower concentration. So because of this, this fifth complex came into the picture, that is ATP synthase, and it will try to balance the charge in both the places. So what it is going to do, it will pull the protons from the intermembranous space and send it towards the matrix side. So during at single time, 2 protons are getting pulled up from the intermembranous space towards the matrix region, and whenever this is happening, when 2 protons are getting pulled up from the intermembranous space towards the matrix, and since it is coming from higher concentration towards the lower concentration, so whatever kinetic energy that is being harvested, it is going to utilize in the conversion of ADP plus PI to ATP. So please remember that for one molecule of ATP formation, we require 2 protons in total. Okay, so this ATP synthase, that is the fifth complex, since the name is ATP synthase, it is going to synthesize ATP. And how it is going? It is doing because of concentration gradient that is being formed between intermembranous space and matrix. This ATP synthase is going to normalize the thing again. It is going to try to normalize the things, and it will just to normalize all the things, or just to balance the charge between these two regions, it will pull the protons from the intermembranous space and send it to the matrix region. And when 2 protons are getting pulled up from the intermembranous space and getting dropped inside the matrix region, there is conversion of 1 ADP to 1 ATP. So that is how ATP are being formed. Okay, so this is this chemical gradient formation. This whole theory is also known as chemiosmotic theory. Uh, okay, and so this was about electron transport system, that how NADH and FADH2 they participate in electron transport system, and ultimately ATP production happens, right? Now now let us just quickly talk about that in total how many ATP molecules are formed through aerobic respiration. So we are talking about aerobic respiration. If you remember, we had talked about anaerobic respiration as well, and in the case of anaerobic respiration, only two ATP molecules are getting released, that too that were formed during glycolysis process only. But here in aerobic respiration, since many types of processes are involved, so let us just write aerobic respiration starts from glycolysis. So this is glycolysis, right? And the next step is acetylation reaction or the link reaction. The third step is Krebs cycle, and ultimately ETS, through ETS, the ATP production is going to happen, right? So during glycolysis, acetylation or Krebs cycle, there will be production. There was production of ATP molecules. There was production of NADH, and there was production of FADH2. Okay, so if you remember during glycolysis, 2 ATP, net gain of ATP was 2, so 2 ATP were released. Okay, and also NADH number was 2 during glycolysis process, and no FADH2 was being released. In the case of acetylation reaction, there was no production of ATP, there was no production of FADH2, only 2 NADH was released during acetylation reaction as well. And during Krebs cycle, in one round of Krebs cycle, if you remember, we had 1 ATP and 3 NADH and 1 FADH2. But since one molecule of glucose releases two molecules of pyruvic acid during glycolysis process, and those two molecules of pyruvic acid get converted into two molecules of acetyl CoA during acetylation reaction, and since Krebs cycle, in Krebs cycle, two molecules of acetyl CoA is going to enter, so this Krebs cycle has to run two times for in if we are considering one molecule of glucose oxidation. Okay, so during Krebs cycle, in totals, when it is running two times, please remember that ATP production is going to get double, right? It is going to be 2 NADH molecules. From one round it was 3, so from 2 round it is going to be 6, and in the case of FADH2 it will be 2 again. Okay, now we need to calculate how many ATP are being released. For that you should know that 1 NADH, sorry, yeah, 1 NADH is equivalent to 3 ATP molecules, and 1 FADH2 is equivalent to 2 ATP molecule. Okay, so let me just write here total. Okay, so in total 2 plus 2 4 ATP, 6 plus 2 plus 2, that is 10 NADH, and 2 FADH2 through whole aerobic respiration process. Okay, so 4 ATP is 4 ATP, 10 NADH, we need 1 NADH is equivalent to 3 ATP, so 10 NADH into 3 is equal to 30 ATP, right? And 1 FADH2 is equivalent to 2 ATP, so 2 FADH2 should be multiplied by 2 is equal to 4 ATP. So in total we are going to get 30 plus 4 plus 4 ATP, in total 38 ATP. So during aerobic respiration we have got 38 ATP, which is far more than what we got in anaerobic respiration, that is just 2 ATP. So with this, aerobic respiration is complete, and I hope you liked the video, and I'll see you in the next video then. Till then, bye bye, take care, stay safe.